Today, I'm going to be talking about the cell cycle, cell growth, and cell division. So, starting the cell cycle, it just starts off with an egg, and then it'll later form a baby. And this just starts off with a fertilized egg, and that fertilized egg has to divide, and keep dividing till it forms the baby. So, we're going to take an overview of mitosis. So mitosis has different phases, as you can see here. It's characterized with prophase, metaphase, anaphase, telophase. So interphase is basically this first one. Then it's prophase and prometaphase, which is kind of like an intermediate between prophase and metaphase. Then we have anaphase and telophase. And then cytokinesis, which is the splitting of the cytoplasm. Acronym is also can be used as IMAT for like all these phases. So we're gonna start off with interphase first. So interphase is actually not really part of mitosis technically. Mitosis actually only consists of prophase, metaphase, anaphase, and telophase. But interphase is part of the cell cycle. And interphase includes G1, S, and G2, and it's 90% of the cell cycle. So its job is actually to produce RNA, synthesize proteins, enzymes, as well as kind of like get the cell ready for mitosis. So the cell has kind of like a life cycle, and that's what we call the cell cycle. So the cell is formed from a mitotic division, and basically, to prepare for this, the cell will grow and mature, and it'll just keep dividing again and again, and go through this loop of G1, S, G2, and M. So there's kind of like two ways, because some cells will keep on dividing, but some cells really don't need to divide that often, and this is kind of the case in like cells in the maybe the liver or the brain. Brain nerve cells, including and muscle cells and liver cells. So they kind of just grow and mature, but they don't really need to divide. So they're in a quiescent phase known as G0. Interphase. So we're going to go more into deep about interphase. Interphase is divided into three phases, G1, S, and G2. G1 is known as gap growth, the first gap growth, and the cell's just doing its everyday job basically. The cell will grow, and it's basically kind of starts signaling to divide. Then we have S phase. S phase is basically DNA synthesis. So basically, the cell will basically duplicate and create another set of chromosome or DNA, and it basically copies its chromosomes. And G2, which is the second gap growth, it'll prepare for cell division, similar to G1, but the cell just grows more and produces organelles of proteins and membranes. So this is just kind of like a visual of interphase right now. So now with our here, like as you can see, the nuclear envelope is over there. It's still visible, and we have the nucleolus, which is just basically the central part of the nucleus. And we can see how like basically chromosomes are getting replicated and proteins and organelles are getting produced. Now we're going to go into S phase. So S phase is, like we said, is when the DNA kind of starts to replicate, okay? So it basically must separate the DNA copies correctly to two daughter cells. And this is kind of through DNA polymerase, to where we're able to create two DNAs, and it uses basically something called a semi-conservative model, to where we're kind of like using this one strand and creating two copies from one strand. And basically, this is just discussed about the error rate. The error rate will be around 1 in per 100 million bases. And these can result to mutations into somatic body cells. And somatic just means regular body cells. Well, gametic or gametes kind of refer to your sex cells, like sperm. So organizing DNA. We know DNA is organized in something called chromosomes. And basically, DNA is just a double helix, and it's just going to be wrapped around histone proteins. Histones are over here. And then it's like kind of like thread on a spool. It's like there's a thread and bead model that, if you can see, like it's basically where the histones or the DNA is kind of wrapped around the histones, and that kind of forms like a nucleosome. So the DNA protein complex is also known as a chromatin, and it's organized into a long thin fiber. So this just shows the different levels. So we'll start with DNA, and then when histones are part of the complex, we can classify this as chromatin. And then basically, when chromatin is condensed, it'll form chromosome. So this is kind of a picture over here. We have like the DNA, then a histone wrapped around it. Then we just have it super coiled, and then later form chromatin and chromosomes when it's completely condensed. We have chromosomes. So after DNA duplication, the chromatin will start to condense, and when it condenses, we'll get a mitotic chromosome. So over here we just have a picture of double-stranded mitotic human chromosomes. So over here, just kind of like the a bit of the more deep into chromosomes. So chromosomes will have a centromere, which is kind of just the central part connecting the sister chromatids, and one chromosome will have two sister chromatids, as we can see here. So also we have something called homologous chromosomes, and homologous chromosomes just basically store the same information. So for example, over here, these two chromosomes are homologous and they'll store the same information, but they'll be kind of from two different sets. So now we're going to go into mitosis. So mitosis is just the dividing the cell's DNA, and it will form two daughter nuclei through four phases, which include prophase, metaphase, anaphase, and telophase. So we'll start off with prophase first. So prophase is basically kind of characterized where the chromatid starts to condense to form chromosome, and our nuclear envelope is also going to start to disappear. So as we can see, the centrioles will also start to move to opposite sides to get ready for metaphase. This includes protein fibers across the cell to form the mitotic spindle. The mitotic spindle is kind of formed with microtubules, including actin and myosin. Also, the nucleolus will start to disappear. So now we'll transition to metaphase. So the transition to metaphase is kind of characterized as basically the chromosomes will like kind of like line in on an equator line, which is like an imaginary line in the middle of the cell. And basically the spindle fibers start to attach to the centromeres. And this will create kinetic cores. Kinetic cores is actually what's going to be attaching it to the centromere. And the microtubule, once the microtubules attach to the kinetic core, basically it'll start to separate, and that's kind of like where we get into anaphase. So this is just called the metaphase plate. That's kind of what the line I was talking about earlier, the imaginary line. Formal terms of the metaphase plate, which is kind of just where the chromosomes will come together. So this is kind of an image from what you'll see maybe through a microscope, a transmission electron microscope. Basically, as we can see, the chromosomes are kind of lined up along a metaphase plate, and they'll start to get ready to, the sister chromatids will start to get ready to go on either side. So now anaphase. Anaphase is kind of signaled by the split up of cohesin, which is kind of the protein that connects both sister chromatids. When cohesin gets cleaved, we will get sister chromatids that are separated on either side, and they're separated by kinetic cores. So they'll move to opposite poles and they'll be pulled at their centromeres, which is kind of the location. The way this happens is actually through motor proteins, and motor proteins such as myosin. They'll walk along the microtubules, and basically that's kind of how it moves to opposite sides. And also the polar microtubules will lengthen, and as they lengthen, it'll start to move to opposite sides. So basically, in anaphase, proteins holding sister chromatids, that's what I said was cohesin. It gets cleaved by separase, which is another protein enzyme that kind of like cleaves cohesin to allow the sister chromatids to get inactivated and split. So this just goes more into the chromosome movement, also known as the Pac-Man mechanism. So basically, kinetic core uses motor proteins to walk around chromosomes along attached microtubule, and it shortens as it gets polymerized because it'll start to split apart and slowly go to opposite poles. So now we're gonna go on to telophase. So telophase is kind of basically where the chromosomes should be at opposite poles after anaphase, and the nucleoli will start to form on both cells, and the chromosomes also start to disperse. This means that they're no longer to be visible under a light microscope. Once the spindle fibers disperse, this kind of signals cytokinesis to begin, and this is the splitting of the cytoplasm. Cytokinesis is kind of specific to animals, and constriction, like for animals, cytokinesis is kind of different than how it happens in plants, because in animals it'll actually there's a lot of actin microfilaments around the equator to form a cleavage furrow, and it'll split the cell into two. It's kind of like tightening a drawstring. As you keep pulling the string, it'll keep like forming that cleavage over here, labeled on the diagram. It'll keep like making more of a cleavage, so it completely splits apart. So as we can see, it's kind of shows mitosis in action. So as we can see, that's kind of like how like an overview of like basic mitosis. So this is just cytokinesis and actin. There's like actually a contractile ring of microfilaments. And it'll just keep depolymerizing. Like that's kind of how the cleavage floral will form. Now cytokinesis in plants is a bit different because we're gonna have something called like a cell plate that's gonna form. The cell plate will form and the vesicles just line up at the equator, which are derived from the Golgi. That's where the cell plate's going to form. And basically, the vesicles will fuse to form two cell membranes, and the new cell wall laid down between the membranes will be kind of like our cell plate. So here we have a picture, kind of like just how the cell plate forms, and that's kind of how we form the two daughter cells. So this kind of just shows the DNA organization and like how like the chromosomes basically duplicate from in mitosis because we can see at G1 we'll start with 46 chromosomes and chromatids will actually doubles from S phase as we can see to 92 and then as we get to mitosis it'll be 92 until we get a cytokinesis where it splits to form 46 chromatids. And the reason the chromosomes stay constant here is because only till anaphase will the chromosomes come apart because technically they're considered as one. So we still have 46 because such a chromatid will just make one chromosome. So DNA content is also similar to chromatids. As we can see, it's like correlates exactly because as we have 2X, we'll also have 92 chromatids. And this kind of just shows like graphs that you might be tested on. 1, 2, and this just shows the amount of DNA. As we can see here, the difference between 1 and 2 is probably between G1 and S because that's kind of when it doubles. And then S is like that number 2 as it starts doubling, and 3 is kind of constant. And then it'll just YV is when it dips, and that's when it dips is probably in between like telophase and cytokinesis. And then V is like the end of cytokinesis. So the evolution of mitosis is kind of through binary fission. Mitosis in eukaryotes probably evolved through this way in bacteria. Now we're talking about the regulation of cell division. So cell division is actually kind of like a very important thing in our body, right? Because if it starts going like uncontrollably, cancer will start to form, which is basically literally just the cells keep on dividing without no regulation. That's why we need a regulation system. So good thing our cell actually has a system. And this is kind of so first cells actually divide by a frequency. So for example, skin cells will only divide 12 to 24-hour cycle and they divide frequently since a lot of like skins like our outer layer like we need to keep dividing for skin because there are a lot of dead skin cells in our body. We need to keep replenish those. Liver cells, on example, we can like reserve it. It will retain its ability to divide. It only divides once every year or two. And we also have nerve cells, which basically never divide, and they're permanently in G0, which is kind of like the space which is out of the cell cycle, where there will be might be subjected to apoptosis. So there's two irreversible points in the cell cycle, and these are also known as basically checkpoints. So the process is assessed and possibly halted at these checkpoints. It'll basically check for something, and if it doesn't meet the requirement, it'll basically either track it back to G0 or it'll halt the cell cycle there. So these are our checkpoints. We'll have a G1, a G2, and an M checkpoint. So at the G1 and S checkpoint, it kind of ask the question, is the cell ready for DNA synthesis? If it is, we'll move on to G2 and M. And over here it asks, has it done correctly? Are there basically no errors? If that's the case, there's no errors, then we can move on to the spindle checkpoint at M. And that's basically, are all chromosomes attached to the spindle? Once they are, then we can carry on and move on to anaphase. So the spindle checkpoint is kind of between metaphase and anaphase. It just checks to see oh, are we able to divide and move to opposite sides? So this is kind of the most critical checkpoint, because if the cell kind of receives a go signal, that means it'll be ready to carry on throughout the most of the interface. So internal signals are kind of like cell growth, cell size, cell nutrition. External signals can be growth factors. If a cell does not receive the signal, it won't able to go past. It'll exit the cycle and go to G0. So G0 phase. So the G0 phase, we're gonna go more into this because this is kind of like this phase where if the cell cycle might be halt, or maybe the cell just doesn't divide like nerve cells or liver cells. It's kind of called the quiescence state of the cell. It's non-dividing, and basically they're kind of like arrested and they'll never be able to divide. So how do cells know when to divide? There are many signals, cell communication signals in the cell. Chemical signals, cytoplasms can give clues. Signals include many mean proteins like activators and inhibitors. So these signals are either can be classified internal signals and external signals. And primary mechanism of control are kind of through phosphorylation and kinase enzymes, and this through like a kind of like a cascade of signals through where like enzymes will phosphorylate something, and like it'll activate a protein kinase like A maybe, and then like through this cascade, signals can carry throughout the cell and kind of like tell the cell when to not go or when to go, or it's like start dividing. CDK is a common enzyme that like actually like literally regulates the whole cell. CDK, and once CDK combines, it can form the mature promoting factor MPF, and that's kind of like allows it to go to M phase when CDK combines with cyclin. So cyclins, like this is similar to what I talked about. When CDK combined with cyclin, will form a mature promoting factor. So once we form this complex, which is also basically just MPF, that, as we can see, MPF activity. Once we form MPF activity, that allows us to go to mitosis, but it'll start degrading as our cyclin starts to degrade. So over here just kind of gives an overview of everything. Our CDK and cyclin is kind of at the M checkpoint, and over here it's G1 and S is more about growth factors. Those are like kind of like the signals we need. CDK and cyclin make up the checkpoints at G2 and M. And over here, there's going to be enzymes that might check for chromosome attachment at the metaphase play. And that specific enzyme is the anaphase promoting complex, or APC. So this is a cyclin expression basically. At G1 phase, we'll start to produce cyclin, but cyclin will start to degrade as we go on later. And that's kind of like how the cell gets regulated. As cyclin starts to form, it'll combine with CDK, and that's what's able to pass the cycle, like the checkpoint. So as we can see, CDK and cyclins will drive the cell from one phase to the next. And this is the proper like regulation we need to prevent like diseases like cancer. So this is just a few signals. Density dependent inhibition. Basically, when we have too much cells crowded, it'll just stop dividing. Because kind of like the name, density dependent, depends on the density. If there's too high of a density, it'll just stop dividing. We also have anchorage dependence. So to divide, cells might be need to attach to a substrate. Only if they're attached to a substrate or like anchor, like basically depends on an anchorage, then it'll start divide. So this kind of just more complex with like other cascading enzymes with E2F that you don't really need to know. It's mainly CDK and cyclin. This is just a growth factor, probably most common growth factor, platelet-derived growth factor. It's made by platelets in the blood clots. And binding of PDGF to cell receptors stimulates cell division. And that's kind of how we heal. So growth factors can actually create cancer, and proto-oncogenes are kind of important because these are normally like they're what normally activates cell division in our cell. If a proto-oncogene basically stops working or something happens to it, it gets mutated, they'll become oncogenes. These are cancer causing when mutated. An example of a proto-oncogene is RAS, and RAS will activate cyclin. Tumor suppressors genes normally inhibit cell division, and if switched off, they can cause cancer, because basically they kind of suppress the formation of tumors or uncontrollable cell growth. Example is P53. So P53 is kind of the most the master regulator as it suppresses tumors. It's like the main. It's the kind of like the called the father or the father of tumor suppressing, and it kind of triggers the destruction of cells damaged beyond repair. Once cells like are damaged and can be repaired, it'll just basically trigger the destruction, apoptosis. We have normal, abnormal. Abnormal P53, as you can tell, it will stop working and the cells just keep dividing and dividing. There's no regulation. Basically, it's just development of cancer. Just a few key points. Angiogenesis will form, the formation of blood vessel growth. It'll escape apoptosis, and that's like apoptosis is a way to prevent cancer by turning off suicide genes, which kind of are important part of apoptosis. Apoptosis, it'll be able to escape apoptosis. It can ignore checkpoints by turning off P53, and this and just cause unlimited growth. So what causes mutations in cells? And these can just be triggered by literally you going out in the sun when the UV is high, UV radiation, cigarette smoke's common one, lung cancer, chemical exposure, radiation exposure, especially if you're in the lab and you don't have proper equipment, exposure to chemicals or even in a high radiation facility. If you don't have proper like equipment, you can easily have a high risk of mutations. So they're just talking about tumors. There's kind of two types. Benign is abnormal cells, but they don't usually cause. They're more harmless. They don't really cause severe problems and can be easily removed. Then we have a malignant tumor, which is more very. That's a harmful tumor, and basically, this is when like there's a risk of metastasis to where the tumor will start to grow. In a malignant tumor, the cells will start to leave its original site and it'll start to be carried by blood and lymph system and start to form more tumors. Over here, this is traditional treatments for cancer. High energy radiation, chemotherapy is common, and through chemotherapy, it will be able to stop vessel growth to inhibit metastasis and stop DNA replication and stop mitosis. That's kind of why also through chemotherapy, a common side effect is balding, and this is just a whole pathway what we kind of discussed about regulation. Mainly, this is way more complex than what we discussed, though. You're just kind of like an overview. Just the main thing to take away from this is how much proteins and enzymes are involved to regulate our cell cycle, and that's regulation of cell cycle. And that's all for today. Thank you.